Today, I'm going to discuss some preliminary results from research conducted by Sally McClure, Doug Blodgett, and myself related to the export of zooplankton and algal biomass from the Emiquon Preserve to the Illinois River during the summer drawdowns. Historically, the site was a highly productive floodplain connected to the Illinois River. The floodplain was disconnected from the river by levees in 1924 to form the Thompson Drainage and Levee District for row crop agriculture, and was subsequently farmed for about 80 years. In 2000, the Illinois chapter of the Nature Conservancy acquired the drainage and levee district with plans to restore not only the wetlands and the lakes and the prairies and forests associated with the site, but also to restore a more natural flood pulse system that resembled the historical hydrologic regime when the site was still connected to the river. In 2016, the water control structure shown in this slide was completed that now can be used to control water exchanges between the preserve, which is on the right side of the slide, and the Illinois River shown on the left side of the slide, and thus helping to restore a more natural hydrology to the restored floodplain habitat. Now, while there are multiple considerations and some concerns related to connectivity, we do have some specific metrics called Key Ecological Attributes, or KEAs, that we can use as guidelines to assess how water management affects wildlife and habitat at Emiquon. The KEAs associated with connectivity are related to sustaining the health and accessibility of the floodplain for species. That includes providing backwater, spawning, and nursery habitat for riverine fishes, hydrological requirements for several vegetation conservation targets, providing moist soil habitat for migratory waterfowl, as well as foraging and nesting habitats for rails, coots, and other breeding birds. Now, these KEAs all focus on maintaining health and access to the floodplain. However, these backwater habitats also provide important food and nutrients to the aquatic plants and animals that inhabit the main river channel. So for the last couple of years, our team has been collecting data to address a KEA that focuses on this transport of secondary production delivered to the river from Emiquon during drawdowns. Now, while primary production relates to the formation of mass for organisms that make their own food, like plants shown here, secondary production is the formation of living massive heterotrophs over time, those organisms that do not make their own food. So our research over the years has included measures of secondary production of aquatic invertebrates that includes insects, microcrustaceans, and zooplankton. Now, these backwater areas like Emiquon are teeming with invertebrate life, from the benthic invertebrates that live in the sediments, many of which emerge as adults and provide food for swallows and other insectivores, and the epiphytic invertebrates that inhabit the vegetation, all important food for fishes, waterfowl, and migrating birds. Today, I will focus on the plankton. These are very small animals that inhabit the water column. This group of organisms are an essential part of any aquatic systems, as all fish are dependent on them at least one stage of their development. They're primary food resources for almost all larval fish of all species, and some species like paddlefish depend on them throughout their life cycle. So zooplankton are a very diverse group of animals that fall into several major taxonomic groups that I'm going to talk about today. Although microscopic, some of the larger groups include cladocerans, copepods, and ostracods. The cladocerans can be found in multiple types of habitats, but many are planktonic. They live in the water column. They have distinctive body plan with two-valved carapace, covers most of their appendages. They're generally transparent, and as can be seen here, they carry their eggs in a protected brood couch covered by that transparent carapace. Copepods are extremely diverse. The common ones that we see at Emiquon are shown here. And in this case, the females carry eggs externally. And when those eggs hatch, they're referred to as nauplii, shown here as well. They're the larval form of those copepods. And you'll hear that again in this talk as well. Now, ostracods are characterized by a body completely enclosed by two valves. Thus, they have a seed-like appearance, and sometimes they're referred to as seed shrimp. Now, another microscopic group of plankton include rotifers. Rotifers are one of the smallest multicellular animals on the planet, and a closer look shows various specialized organ systems. To date, almost 100 species have been identified from Emiquon. We also measured algal biomass by extracting chlorophyll A, a pigment that absorbs light energy and is stored in its chloroplast. It can give us a measure of the algal biomass. Just for some context, we've been monitoring the zooplankton Emiquon for many years, prior to and after the restoration. This slide shows a snapshot of the zooplankton community composition at Emiquon. As you can see here, rotifers dominate in terms of abundance. They generally comprise about 80 to 90% of total zooplankton abundances. This hydrograph shows water surface elevation through time at Emiquon from around 2003 to 2021. And the water control structure then was completed in 2016. And since then, there have been several drawdowns using gravity flow in 2016 and 2017, and that's shown by those red circles. In 2018, 2020, and 21, low water levels resulted from using the structure's 2 electric pumps, and that's designated by the green circles. Our previous sampling years are designated by the blue squares. And then our current work focuses on the last 2 drawdowns in 2020 and 2021. So this aerial of Emiquon shows Thompson and Flag Lakes and the pump station, which is the red circle on here, where the control structure is located. During the drawdowns, we collected water leaving through the control structure to measure export, and our sample sites here are shown as those yellow circles. We also sampled at 4 sites in the pumphouse ditch that included a site at the pumps and 3 additional sites heading east towards Thompson Lake. And then we also had a sampling site in the lake itself. We sampled weekly during 2020 and shorter sampling intervals every 4 to 6 days or so in 2021. Zooplankton were collected using a diaphragm hand pump, and this was connected to a weighted hose that could be lowered and raised to collect samples from the entire water column. At each site, we measured basic field measures shown here: dissolved oxygen, temperature, etc., at the surface, and then also near the sediments at the bottom at each site. We also did Secchi transparency and collected water samples for chlorophyll A, total suspended sediments or solids, total nitrogen, and total phosphorus. We also collected water samples for the same analyses from Illinois River and near Havana. All right, so in the 2020 drawdown, the highest abundances of zooplankton were exported from Emiquon to the river in June, numbers ranging from about 4,200 individuals per liter to over 13,000 individuals per liter. The export stabilized in July and August, averaging around 2,300 individuals per liter. Looking at the taxonomic groups making up those abundances, we see that rotifers comprise most of the abundances exported from Emiquon, generally making up 70 to 90% of total abundances on any given day. Now, again, this is not a surprise, given that we already know that rotifers comprise 80 to 90% of the zooplankton community from our previous research. We do, however, see some cladocerans early in the season, shown by those dark gray bars, and this kind of consistent persistence or consistent presence of copepods throughout the drawdown. Now, biomass leaving the site follows a similar pattern as abundances, with highest biomass early in the drawdown, and then an average of around 150 micrograms of dry mass per liter in July and August. So the next few slides focus more closely on the taxonomic groups that contributed to the export of zooplankton biomass from Emiquon. The slide shows biomass on the right axis and the relative abundances of cladocerans, copepods, and rotifers on the left axis, according to the legend that's shown at the top right side of the slide. So the big pulls of biomass in the first 2 weeks of June were driven primarily by cladocerans. They made up about 40 to 65% of the biomass leaving the preserve. Now, at the end of June, biomass export was primarily comprised of rotifers, shown by that open bar here, making up about 60% of total biomass. Now, rotifers remained relatively important component of the biomass throughout the summer and peaking again at the end of July, making up about 75% of the total biomass exported from Emiquon to the river. Copepods remained a steady source of biomass export throughout the summer, ranging from 20 to 73% of total biomass exported. If we look a little closer at the cladoceran community, the timing of the drawdown really captured a seasonal shift in the cladoceran community that agrees with our longer-term annual monitoring data. The early pulse of biomass to the river was mainly comprised of native cladoceran species, those being Bosmina, shown on the far left, Chidoris, down here at the bottom with the dark bar, and then this native Daphnia species, Daphnia galeata, shown on the left. A community shift generally does occur in midsummer as these native Daphnia species decline. And then we have a non-native Daphnia species, Daphnia limholtzi, shown on the far right here, that begin to emerge later in the summer. And other species like Diaphanosoma are also more abundant later in the summer, comprising the majority of the cladoceran biomass. However, these are very small individuals. And so even though we may have high numbers, they have smaller biomass. And if we add rotifers into the mix, although we have more than 100 rotifer species that we've identified from Emiquon, a few genera dominate the biomass of the system in any given season. So during the summer drawdown of 2020, Polyartha, shown here, comprised 30% of the total biomass, total zooplankton biomass exported in late June. Several species of Branchionis averaged about 20% of total biomass in July, followed by a pulse of Splankna. This is a really large rotifer. It's actually a predator and consumes other rotifers. In this particular specimen, you can see several Caratella, several other rotifers inside this Splankna. So even though they're microscopic, rotifers are a really important component of the biomass exported from Emiquon to the river. So copepods, like we've been seeing kind of throughout this talk, they remained a steady source of biomass export throughout the summer, ranging from 20% to 73% of total biomass exported, and generally averaging around 41% during the drawdown. It's important to note that the highest biomass of these copepods occurs in July and August, when the abundances of the larger bodied cladocerans have declined. Thus, the copepods, they provide a steady food resource throughout the summer and are especially important later in the summer when other food resources are less available. A little look at the algal biomass. In both 2020 and 2021, algal biomass exported to the river, which is shown in green, very much aligned with the average algal biomass that was measured across the sampling site within the preserve, and that's shown by these open circles. And both of these were consistently higher than those measured from the Illinois River in Havana, shown as those solid circles. And we ran a quick t-test on these data. We show that the mean algal biomass exported from Emiquon during each of the drawdowns was significantly higher than that in the river. So in summary, we know from previous research that the invertebrate community that includes the zooplankton are productive at Emiquon and provide an important food source for the aquatic and terrestrial communities. Now, this study shows that Emiquon can also contribute both algae and zooplankton resources to the larger river system. Zooplankton export from Emiquon likely subsidizes food resources for riverine species, contributing between 1,300 and 13,000 zooplankton per liter to the river over the 2020 summer drawdown. Now, given that the net reduction in water volume during the 2020 drawdown was about 4.7 billion gallons, this is a lot of zooplankton and algal biomass delivered to the river. Now, we do realize that some of these reductions were due to evaporation and transpiration, but even so, what left the preserve through the control structure are extremely high numbers of zooplankton. Now, with the introduction of filter feeding carp, that voracious appetites, voracious filter feeders, this is especially important for our native filter feeding fish species like paddlefish. And in summary, so these drawdowns can provide the ability of these backwater areas like Emiquon to perform this historical essential function of providing high quality food resources to the larger river system. With that, I thank you very much.